Okay, thank you everybody for coming today, and thank you to all of those who are online. It's an absolute delight to be able to showcase some of the research that's going on in the Centre for Inflammation Research and Translational Medicine, or CIRTM as it's favorably known. The point of today really is for our younger members of the Centre, so our postdocs and our PhD students, to really show you what exciting research they've been doing. So the agenda for today, I'm going to do a little introduction to the Centre, and then we'll have talks from our students and postdocs, and then we're going to leave this room and we're going to have a look at some of the posters. I should say there's also going to be some prizes for the best oral communications and posters. We also thank, we've got PCR Biosystems that are outside that have sponsored this event, so please do go and visit. Have a chat with them because they've generously donated funding for the lunch pizzas, for example. So yeah, we're very grateful to them. I personally just want to say a special thank you to Jackie and also to Keith, who are academic members of the Centre. Both of them have been absolutely instrumental in putting today together and doing all the running around and getting people, coordinating people and their talks and posters. So a personal thank you from me, in particular to both Jackie and Keith. Okay, so a little bit about, just for a couple of minutes, a little bit about the Centre. I'm Felicity Gammins. I'm a Professor of Pharmacology here at Brunel, and I'm Director of the CIRTM. The Centre was set up in October, roughly, 2019, with the objective of bringing together a critical mass of both basic scientists and also clinical scientists tackling today's global health challenges. We want to promote, and we're driving toward promoting interdisciplinary and multidisciplinary research to provide an outstanding environment for both the research and the research training. So we're training the next generation of scientists, and that's very much something we're very passionate about within the Centre. We're very lucky to have funding from all sorts of charities and funding bodies, without whom we wouldn't be able to do a lot of the research that we undertake in our Centre. So we're extremely grateful to them, and grateful that they recognise the importance of the work that we're doing within the CIRTM. I've mentioned that we tackle a number of global health problems. Those are, for example, cardiovascular diseases, inflammation, which is a key driver of multiple diseases, infections and immunity, cancer, microbial resistance, and microbiome and health. We focus on these because they affect billions of people, not just across the globe. And so we really want to understand further the complexities of these different disease states in order for us to be able to help society. And you'll hear about some of the research that we're doing in these areas as we go through today. We tackle these challenges in different ways. We've got state-of-the-art techniques. Some of you are going on lab tours later, so you'll be able to see some of the methodologies and the instruments and the techniques that we use. We also use different systems, so we all the way from bacteria up to humans, taking samples and using those as models of these diseases in order for us to understand the complexities of these challenges. We do that, we work across different dimensions to obtain an integrated understanding of health and disease, and by that we go from the genes all the way up to the population network. Hello. Including sort of molecular, cellular specific effects, and then organ specific effects. And you'll hear about some of these today, and obviously we're very happy to discuss more outside of this. We interact both across the university with different disciplines, computer sciences, for example, AI, the importance of AI in the research that we do, to social sciences, social dynamics, behaviour, for example. And outside of the university. So there's a very strong interdisciplinary and multidisciplinary and cross-disciplinary approach to our research. These are the academic members at present. We're not going to hear from any of them. We're going to hear from some of their postdocs and students. to talk about their research today. So that's it from my side of things, other than it's an absolute pleasure to be able to hear about the research that's going on in the centre, and I hope that you'll be as enthusiastic about it as I am. And so without further ado, we're going to start off with Sophie Howard, who's going to talk to us about the evolution of native biofilm communities on waste plastic to enhance polyester degrading activity. Sophie, it's over to you. Thank you very much.
Okay, thank you for offering me a talk for today. So I'm a postdoc in Ronan McCarthy's lab. Work on something a bit different to a lot of you guys, but obviously plastic degradation and plastic waste is a huge human health problem as well, not just an environmental problem. Okay, so just an introduction into what the plastic waste crisis is. So really, mass production of plastic started in around the 1950s, when 2 million metric tons of plastic was produced annually. And in 2017, it was recorded as 8.3 billion metric tons of plastic produced in total, which at the moment is equal to about 400 million metric tons per year. And that amount per year is just increasing each year, even with the increase in reducing single-use plastics or using bioplastics and stuff. It's still predicted that by 2050, there will be 34 billion metric tons of plastic that's been produced. And that's just in 100 years space. So of this total plastic that's been produced, this is from 2015 as well. So this is already 8 years old, this data, but there's 6.3 billion metric tons of plastic waste. So by now, that's equivalent to about 1 billion elephants worth of plastic waste. And by 2050, it's predicted that there will be 12 billion metric tons of plastic waste in landfill and the environment. So that's not even including what goes to recycling. So currently, globally, only 9% of plastic is recycled, which is also a really shockingly low number. 12% is incinerated, and whilst this does remove plastic waste completely, and it can generate electricity, it also can create carbon emissions and toxic fumes, which is obviously bad for human health. 79% goes to landfill, which is obviously really bad for the environment, and there's hundreds of toxic and cancerous chemicals in plastic, which leaches out into the soil and water. So whilst recycling is great, it has its limitations. So the quality of plastic reduces after recycling, and plastic can only be recycled a few times and then it's just no longer recyclable. In general, it's also more expensive than making virgin plastic from petroleum sources as well. So we really do need an alternative source solution to go alongside recycling that we can replace landfill with. So our potential solution is plastic biodegradation by bacteria. So essentially, it's been known for a few years now that quite a few species of bacteria can actually degrade plastic. And they do this by essentially eating away at the plastic surface and using the breakdown products for energy. So there's obviously a massive amount of plastic waste in the environment where bacteria are exposed to that plastic. And a recent study found that the presence of plastic degrading genes is strongly correlated with the presence of plastic waste. So the fact that we've been polluting the environment with plastic has driven the evolution of the bacteria to be able to degrade the plastic. So we hypothesized that if there's plastic waste, there's likely to be bacteria that can degrade plastic in the vicinity. So to actually be able to use this as a solution to plastic waste crisis, and actually manipulate this natural resource to develop a biotechnological tool that can be applied in industry to degrade plastic on a large scale, we need to tackle the issue of degradation efficiency. So many environmental bacteria that can degrade plastic do so at a really low efficiency, because in the environment, they've got alternative carbon sources they can use for energy. So we really need to improve this efficiency. So many research groups are actually looking at mutating enzymes specifically to actually improve their efficiency. And this is working really well, but we wanted to look at community evolution instead. So we actually want to isolate plastic native communities and then evolve them as a whole. So our aims, firstly, based on the fact that we think plastic-associated bacterial communities should be more likely to be able to degrade plastic, we want to isolate native bacterial communities from plastic waste and then assess and characterize their plastic degrading potential. And secondly, since the degradation efficiency is often quite low, we want to evolve native bacterial communities to have optimized plastic degrading activity, as well as characterize the evolved communities as well. Okay, so firstly, because native plastic associated communities are more likely to have plastic degrading bacteria, we need to find which type of plastic actually promotes the most bacterial biofilm to start with. So we did a biofilm assay on different plastics, and we just did this in the lab with a coli strain that forms high biofilm. And we found that expanded polystyrene, which is EPS, the one on the left, promotes the most biofilm formation. So this seems like a good candidate to isolate large amounts of plastic associated communities. So we sought out EPS waste. Specifically, Ronan collected some EPS samples from the coast of Ireland, and then we performed an evolution experiment on it. So each piece of plastic waste, which we had 16 pieces, was split in two. The first half was inoculated into an N9 minimal media culture with no carbon source other than the plastic itself. And then 2 months later, we collected the community, and then this was our evolved community. And then the other half of the plastic was inoculated into an LB overnight to collect the community, and this was the original inoculum from the plastic. And then to assess their plastic degrading activity, we did a PCL assay. So PCL, or polycaprolactone, is a model polyester type plastic, and we can dissolve it in agar. So we can do a really nice screening assay where we end up with cloudy agar, and then when the PCL is degraded, the agar becomes clear, so we can actually measure the PCL degradation. So after the evolution experiment, 7 of 16 plastic associated communities were able to degrade PCL. So to start with, we did have the occasional amount of PCL degradation, but it was really weak. So this graph just shows the dark blue is the size of the bacterial spot on the agar plate, and the light blue is the zone of clearance that forms around it. So that's how much degradation of PCL there is. So for example, PS13, which is in the village, the original community could degrade PCL, but it was really faint on the plate. And actually, when we plated out the whole community on PCL agar plates for single colonies, only 4.8% of those colonies could actually degrade PCL. So it was actually a really low number of the community who actually had the ability. And then once we did the evolution experiment, you can see here we've got these zones of clearance around the colonies. 94.7% of the evolved community was able to degrade PCL. So we have a vast improvement in the community for evolution and for degradation of PCL. So to just characterize, specifically we focused on PS13, we did 16S metagenomics of the evolved community, and we found that the majority of the species was actually Pseudomonas stutzeri. So Pseudomonas stutzeri is quite a common environmental isolate species, and it has previously been linked with degradation of polyhydroxyl butyrate. However, this is a bio-derived plastic rather than a petroleum-based plastic, which is what we're looking at for degradation purposes. Additionally, several Pseudomonas species have been linked with plastic degradation before as well. So this adds to that. So we isolated AP stutzeri from PS13 to continue characterizing the particular strain. We found that it could grow using PCL as a sole carbon source. So you can see in this growth curve in pink, the only carbon source is PCL, and it does increase in OD over time. And then in green, when there's no carbon source, the species does slowly degrade and decrease in OD over time. And this actually resulted in weight loss of PCL. So it is actively degrading the PCL and causing it to lose weight over time. So we also did whole genome sequencing of this P. stutzeri isolate, and we found that it contained 2 polyesterase homologs and 1 N-hetase homolog. So both of these enzymes are involved in PET degradation. So the PETase or polyesterase degrades PET into BHET, MHET, and TPA. And then MHETase can further degrade MHET into TPA and ethylene glycol. This last step is optional. Not all bacteria actually have both enzymes. Most just have the first enzyme. So it's likely that one or all of these three enzymes are actually involved in the PCl degradation we've seen, and that'll be future work. Specifically, we found that one of our homologs, PSP1, was a really good match. It had 82% identity to the polyesterase PMC from Pseudomonas mendecinum. So this one is particularly promising. So to conclude, we found that community evolution experiments can be used to improve the plastic degrading efficiency of environmental bacterial communities. The selective pressure of having only plastic waste as a carbon source drives the community towards strains with good plastic degradation. We found that Pseudomonas dozeri became the dominant species after evolution, and it was able to degrade PCl and use PCl as a sole carbon source. And that it likely does this using some or all of the three putative polyester degrading enzymes that we found in its genome. So the future work will be partly to characterize these enzymes, confirm they are functional and that they can actually degrade the PCl that we're seeing, and see if once they're purified, they actually also degrade PET as well, which is a harder to degrade version of PCl. We will also use this method again on other plastic waste to assess the evolution of other bacterial communities and see what else we can find in case we find some more novel bacteria or novel enzymes on these plastic associated communities. And this is all working towards making efficient plastic degrading bacteria. And then once we have efficient plastic degrading bacteria, we can then actually use this in industry to go alongside recycling to combat the plastic waste crisis. And I think that is everything. Yeah. So just to thank everybody who's involved in the work and our funders as well for Horizon 2020.
Super. Thanks very much, Sophie. That's great. Okay, our next speaker is Akua, who's going to talk to us about the influence of daily temperature and the impact of malaria incidents in Ghana. Thank you very much.
Thank you. Thank you all so much. So after having the pizza, I went to the other venue where we met last year, and I was thinking I was so early, feeling proud of myself and wondering why everyone else wasn't there. Later, I realized that I was lost. And so I'm grateful I finally found and remembered the room. My topic is examining the influence of daily temperature variations in measuring the impact of climate change on malaria incidents in Ghana. This is just a part of my work. Broadly, I'm looking at malaria being a climate-sensitive disease, and I'm examining that part in Ghana. And so malaria is one of the world's longest, oldest inflammatory disease, but in a highly scientific audience, I dare not try to explain how malaria is an inflammatory disease, but you all know. And so it's a serious issue still in Africa. In every minute, a child dies from malaria in Africa. And so that makes it a very important disease to look at. Findings of temperature, rainfall, humidity as the most impactful climate variable when it comes to malaria transmission. And so I'll be looking at all the three, and then I would also add wind speed, probably wind direction, and then sunshine. But I'm at the point where I'm investigating the influence of temperature. And at this point, I realized that a lot of laboratory and modeling studies are offering a varying range of optimum temperature between 15 to 40 degrees. Some studies offer 28 degrees. Some studies, according to their model, they come out with 25. Other studies, 26. And so within 15 to 40 degrees, we have varying range of temperature that the experiments offer as the most optimum temperature for transmission. And so, though it gives the baseline that temperature is highly correlated to, or temperature impacts malaria greatly, the discrepancies in the transmission threshold is critical for the interpretation of the risk involved. That was according to Mizan. And so I'm looking at trying to account for what most of the studies actually uses either minimum temperature or maximum temperature to talk about how malaria is being influenced. But when you do the analysis, you realize that between minimum temperature and maximum temperature, there is the influence of daily temperature variation. And a lot of studies are not accounting for it. But we know that as ectothermic as the anthropod or the mosquito vector is, it is definitely being affected by the daily temperature variation. And so there is a cause for people to overestimate the impact of temperature on malaria transmission or underestimate the influence. And so I identified this gap. Currently in Ghana, no one has done studies that account for the daily temperature variation. So what I'm doing is, these are the 22 synoptic stations in Ghana. They report weather conditions, and so they have data for temperature, maximum temperature, and minimum temperature. I'm using our programming in Microsoft Excel. I find the daily transmission, the diurnal temperature range by subtracting the minimum temperature from the maximum temperature. And then I'll use time series analysis, correlational analysis to identify or account for the impact of daily temperature variation on malaria. And so currently I don't have my malaria data, I have the climate data. That's what I've spent time on to analyze. And when you look at this figure, you realize that ordinarily, if anybody was using the minimum temperature to talk about how malaria impacts, how temperature impacts malaria, the person would have used 21.22 for January, the mean minimum, or any of these figures. I mean, when you're doing your analysis. But then, when you subtract the maximum, and if you're using maximum alone, these are what you're going to do. These are the figures you're going to do. But when you subtract the maximum from the minimum, what happens throughout the whole day is that these figures are not accounted for. So I am trying to investigate how this influences. I've also done box plots for all the ecological zones. Actually, Ghana is divided into 3 ecological zones: the climates, the coastal climates, forest climate, and then savanna. And so I did analysis based on the ecological zones, and I realized that the threshold or the ranges are similar among the ecological zones. And so this is also one of the things I'll consider when I get my malaria data, to understand how the difference in ecological zone varies when it comes to the impact on malaria. And then also I have done a time series analysis, and then I realized that this is the incidence of malaria mortality in Ghana, and an annual incidence. Okay, but I kind of find a similar pattern where there is a decrease from 2000 to 2005. You see that here, and then it increases later, and then comes down again. So I'm also going to, when I get my malaria data, I'll also factor time series analysis in the correlation between diurnal ranges and malaria case. So to the best of our knowledge, this will be the first epidemiological study that explores the relationship between diurnal temperature ranges and malaria case rates in Ghana. And it would also form the background for understanding and predicting future changes in malaria incidence in Ghana. Thank you.
Thank you very much. Okay, so that's the talks from our PhD students, and now we're going to move on to our postdocs. I'm going to hand over to my co-chair, Jackie.
Okay, good afternoon, everyone. I'm Jeff, and I'm a postdoc with Professor Arturo. And I've been looking at therapeutic effects of hemolytic stem cells, genetically modified, on Friedreich ataxia disease mouse model. Okay, so disease predominantly affects the sensory neurons, cardiac cells, and beta cells. It affects 1 in 29,000 of the Caucasian population. And it's occurs early on in life, around 5 years old, and patients tend to live up to around 40 years old. The symptoms include movement dysfunction, so abnormal gait, reduced reflex actions, and heart dysfunction. And in the majority of patients, it results in heart failure as the primary cause of death. Okay, so what's the cause of the disease? And it's due to reduction in frataxin FXN levels, and this is due to a mutation in the frataxin gene. There is about 90% reduction in cellular frataxin levels in these patients. So obviously the therapeutic approach is to restore cellular frataxin levels in patients, and we want to achieve this by using genetically modified patient HSCs, stem cells that secrete a cell-penetrating fusion peptide, frataxin fusion peptide. And this is the plasmid which we want to incorporate into a lentiviral vector in order to get it into the cells. Okay, so the aim of the study was to look at the therapeutic effects of these genetically modified HSCs secreting the fusion protein in the YG8 LR and XLR mouse model of the disease. So first, in vitro studies, we infected HEC cells and fibroblasts from patients to see if we can get production of the fusion protein. So firstly, with the fibroblast cells, the infected cells, as you can see, produce the mature form of the protein, whereas those uninfected, we cannot detect anything. Whilst infection of the HEC 293 cells, and looking at the culture media of these cells, we see the immature frataxin in both the infected cells, but not in the uninfected. So this would show that the fusion protein is being secreted. Okay, so the next question is the secreted protein getting into our target cells? As you can see, these are the healthy fibroblasts, and those stained in green are the frataxin, whilst those fibroblasts from frataxin Friedreich ataxia patients, you see no green or very little green staining for frataxin. But when we treat this with the supernatant containing the fusion protein, you can see there's bright green, so it's managed to go into our target cells. And again, the controls with the empty vector, we don't see any frataxin increases. So the protein itself is important for mitochondrial activity, and this can be measured using the aconitase activity assay. As we can see, when we treated the fibroblast cells from patients with the fusion protein, there's a dramatic increase in aconitase activity levels compared with the empty plasmid treated cells. So great, it's producing protein and it's getting into the cells, and it seems to be having an effect on the mitochondrial activity. So now we want to show that the protein itself is able to hit the mitochondria within the cells. So we stain for frataxin in green and in red mitochondria. And as you can see, there's co-localization of the protein within the cells by immunofluorescence. So it's hitting our target mitochondria in these cells. So having shown that, we went on to do the in vivo studies. And this was the plan. We took our Friedreich mice. We took male mice as donors using their bone marrow cells. We enriched for the HSCs using magnetic cell sorting. We then infected these cells with the lentiviral vector, having the fusion protein plasmid. And we injected these into irradiated female recipient mice, which hopefully led to a reconstitution of the recipient mice with male blood cells that are secreting the fusion protein. So these are the groups. First group, Friedreich ataxia mouse, given the HSCs infected with the empty vector. Second group, HSCs infected with the fusion protein vector. And FRD mouse just given wild type HSCs from C57 black 6 mice, and obviously the untransplanted controls. So we used the female recipients and male donors so we can probe for the Y chromosome. This would tell us engraftment levels. So if we do a PCR for a Y chromosome DNA. And we transplanted at the pre-symptomatic stage of disease in these mice, so at about 8 weeks old. So first, we wanted to see that we got production of the fusion protein in these transplanted mice. And when we look at the blood, we can see that there's an increase in frataxin levels in the blood of the transplanted mice with the fusion protein, both at 8 weeks post-transplantation, also at 12 weeks post-transplantation. So it looks like we're getting production of the fusion protein in these transplanted mice. So next we looked at symptoms of the disease, see if we can alleviate them with these with this treatment. So we looked at the activity levels of our mouse. We did the accelerating rotor rod test and also the BMORC tests, all of which show motor function of our mice. So first test, the activity. As you can see, when we compare the black 6 wild type controls, the normal mice versus the Friedreich mice in green, by 16 weeks, you saw a massive decline in activity levels in these mice. However, those that were treated with the stem cells did not show any significant declines compared with the wild type yet. This is also the case for the empty HSC transplanted mice, showed significant declines. However, by 20 weeks of age, you can see that all mice show decline in activity levels by this time. And the rotor rod test, again, we see declines in the diseased mice in green by the time they get to 28 weeks old. However, all those transplanted with stem cells actually did okay. They did not show reduction in rotor rod performance levels up to 36 weeks of age. And finally, the BMORC test at 24 weeks, you see that, again, the diseased mice, the untreated mice, show reduction in performance compared with the wild type normal controls. However, those given the HSCs were protected, and those given the fusion protein HSCs doing the best, remaining symptom-free in this term by 36 weeks of age. So next, we wanted to see whether how well the cells engrafted in our recipient mice, and as you can see, doing the Y chromosome DNA PCR, we did get some engraftment, but not very good. At best, we managed to achieve 10%, just over 10% engraftment in one of the mice, so quite disappointing. And this might be the reason why the therapeutic effects was transient. So quite disappointing. And then we then looked at the pathology of our study mice. As you can see, if we look at neuron size in the cerebellum of these mice, with the black 6 normal mice, there's normal, large, healthy neurons. But in the YG8 Friedreich ataxia mice, there's a reduction in the size of these neurons. And this is also seen in the empty plasmid infected control. However, those given the fusion protein plasmid or vector, as well as those transplanted with normal HSC, the size seems to be maintained at 37 weeks old. And if we measure these, again, we can show that those given the frataxin fusion protein HSCs were protected from this reduction in dentate nucleus neuron size at 37 weeks. So from this, we found that transplantation of this fusion protein possessing HSCs increased frataxin levels in recipient mice, and they had beneficial effects in terms of symptoms and neuronal health. We measured also aconitase activity in these mice, and it increased, and frataxin levels within the heart and brain and muscle tissues of our treated mice also increased. But again, as I said, the transplantation seems to only have temporary effects, and this is due to probably inefficient transplantation engraftment. So with this in mind, we wanted to repeat the experiment, this time trying to optimize the depletion of recipient mice. And I used a brand new X-ray irradiator to try and optimize this. As you can see, we now managed to achieve our target of over 50% engraftment in all of our mice. And I'm now carrying on to perform the experiment again. And that's it. Thank you for everyone in Arturo's lab, Paolo Bagneri's lab, and all the people at ICH for helping us with this study. Thank you.
Good afternoon, everyone. For those of you who don't know me, my name is Kavita, and I'm currently a third-year doctoral researcher in Dr. McCarthy's lab. And my research primarily focuses on drug discovery and colistin resistance in the bacteria Acinetobacter baumannii. But I'll go into a little bit more detail about why this bacteria is a scary, dangerous pathogen a little bit later on. But as for now, allow me to introduce the title of our upcoming paper that we're extremely excited about, really proud of. It's targeting iron as a means to potentiate colistin. So just a little bit of background about the field that I'm working in. So in 2019, 4.95 million people died from an antibiotic-resistant related infection. Now, these are big, huge, scary numbers, but it's largely attributed to this bacteria, Acinetobacter baumannii. So this bacteria is a gram-negative opportunistic pathogen, which is currently resistant to all available antibiotics that we have. And this impressive statistic has put it at the top of the World Health Organization's priority pathogen list for pathogens that are in urgent need for novel therapeutics. So this is where my project comes in. So what we started off by doing was screen hundreds and hundreds of plant compounds, their phytochemicals, against A. baumannii in the presence of the sub-MIC of colistin. Now, this is a sublethal dose of colistin that's so low that if you use it on its own against the bacteria, it has no effect against the growth of the bacteria. So from this initial screening, we identified our top hit potentiators, and Camphorol came out as number one. And it's actually derived from strawberries, a bit of a fun fact. And we've taken this phytochemical all the way from the initial testing, right the way down to identifying its mechanism of action and even testing it in vivo. So our first question after identifying Camphorol was, how effective is this compound? So here we have MIC data where we've tested various amounts of Camphorol in combination with the sub-MIC of colistin, and we found that the most effective dose was 0.375 millimolar of Camphorol in combination with colistin, and it was able to completely inhibit visible growth. But we know that it works, and it works well, but how is it working? So in order to identify its mechanism, we then went on to conducting RNA-seq analysis. So just to simplify this, we found a upregulation of 118 genes and a downregulation of 19 genes. And of the downregulated 19 genes, they were associated with iron storage proteins and iron-dependent enzymes. So this downregulation kind of gives us the idea that the bacteria is trying to save up as much intracellular iron as it can by preventing its usage in iron-dependent enzymes and also preventing it to be stored away. And at the same time as this, we have a upregulation of iron acquisition systems. So these are siderophores that are able to go out of the cell, grab the extracellular iron, and bring it back into the cell. Now, this suggests that there is a limited amount of iron within the cell, and the cell is trying to compensate for this by bringing more iron in. But then what happens if we try and rescue the cell by adding more iron to the media? So this is where it gets a bit interesting, where we found if you add Fe2+ back into the cell, there's no rescuing the bacteria. But if you add Fe3+, you're able to rescue the bacteria and after it's been treated with the combination treatment and restore the growth. So if you take this data combined with the RNA-seq data, you reach this hypothesis. So on this side over here, on the blue side, we have a happy, healthy bacteria cell where you can see the sub-MIC of colicin coming in. And the cell is able to cope with this using mechanisms such as using the SOD-B enzyme, which will take the byproducts of colicin, which it produces when it enters the cell, and detoxify it into less harmful products such as oxygen. And at the same time, there is an equilibrium of Fe2+ and Fe3+, and the cell is able to cope with the ROS being produced, and it's happy. But then on the other side here, we see camphorol coming in. We see it binding to Fe3+, and this is creating a disruption in the iron homeostasis, whereby the cell has to compensate by taking Fe2+ and converting it to Fe3+, and by doing so, it's pushing the reaction towards the Fe3+ and producing harmful ROS byproducts. And at the same time as this, the SOD-B enzyme isn't able to detoxify the sub-MIC of colicin coming in because its cofactor, Fe2+, is no longer available. It's busy being made into Fe3+. So then just to validate our hypothesis, we measured the ROS levels. So over here on this bar, you can see our positive control, which was a lethal dose of hydrogen peroxide. And you can see it produces a significant amount of ROS. Over here, you can see the sub-MIC of colicin, as we hypothesized, produces a increased level of ROS, but the cell is able to cope with this. Camphorol on its own does not increase ROS, but as soon as you combine the camphorol and the sub-MIC of colicin, you can produce ROS levels that are equivalent to the positive hydrogen peroxide control. We then went on to validating the SOD-B enzyme mechanism that we hypothesized. And to do this, we used a transposon mutant. So what this means is the mutant doesn't have a functioning SOD-B, which is equivalent to the cofactor not being available and the SOD-B enzyme not being able to work. And we found that this mutant was highly susceptible to the sub-MIC of colicin, if you compare it to its wild type. And this is my favorite part, where we tested our combination therapy against actual clinical isolates of baumannii, which are highly resistant to colicin. So they have an MIC of colicin of 256 and micrograms per milliliter. And we added just 0.05 millimolar of camphorol and found that we can reduce the MIC from 256 all the way down to 4 micrograms per milliliter, which is amazing. We then went on to testing our treatment in vivo and found that not only are the components of the treatment not cytotoxic, but also you can if you inject the Galeria with a lethal dose of baumannii, you're able to provide a probability of 60%, which makes for some extremely happy Galeria. So just to summarize, Camphora works synergistically with the sub-MIC of colicin, both in vitro and in vivo. The mechanism of action occurs through the disruption of iron homeostasis. And overall, targeting iron bioavailability could be a potential therapeutic strategy to treat colicin-resistant baumannii infections. Just want to say a huge quick thank you to Ronan and Ruben, who's currently not here, and of course everybody in the McCarthy lab for putting up with all the moaning that came along with this project. Thank you.
Thank you very much, Peter. Lovely talk.